Good morning, everyone, and welcome. I am pleased to have you all here today. We will be discussing the qualification of highly purified water for HPLC analysis. The title of our presentation is Dr. S.K. Singh, Ph.D., and today we will explore the key aspects of water quality required for accurate and reliable HPLC results. Over the course of this presentation, I will cover the importance of water purity, the testing methods, and standards to ensure your water meets the necessary criteria for HPLC. So, let's get started and delve into this important topic. Let's talk about why maintaining water purity is so crucial in our analytical processes. Any impurity in water can lead to several issues, such as baseline noise, which can obscure our signals and make interpretation more difficult. It can also cause ghost peaks, those unwanted signals that interfere with our analysis. Additionally, impurities can reduce the lifespan of our columns, leading to more frequent replacements and higher costs. Most importantly, contaminated water can result in non-compliant analytical results, risking the validity of our data. This concern applies broadly across various settings, including QC laboratories, HPLC, UPLC, and LC-MS analyses, as well as GMP and GLP facilities. Now, let's focus on highly purified water, often called HPW, which is a critical reagent in many aspects of our work. We use HPW for preparing mobile phases, diluting samples, creating standards, and system flushing. Each of these steps depends on water of the highest purity to ensure accuracy, reproducibility, and compliance in our analytical procedures. In essence, maintaining water purity isn't just about cleanliness. It's about safeguarding the integrity of our entire analytical process. Let's take a closer look at the regulatory references that guide us in water quality and pharmaceutical manufacturing. First, we have the USB, or United States Pharmacopoeia, which provides specific guidelines like USB 1230-1 for water for pharmaceutical purposes. This section outlines the standards for water used in drug production to ensure safety and consistency. Then, there are the USB monographs that cover purified water and water for injection, both critical for different stages of pharmaceutical processing. Moving to Europe, the European Pharmacopoeia, or EP, specifies requirements for highly purified water, emphasizing purity levels needed for sensitive applications. The WHO Technical Report Series, or TRS, offers comprehensive guidelines on water systems, helping ensure safe and reliable water infrastructure worldwide. Additionally, the ICH guidelines, Q7, Q8, and Q9, focus on good manufacturing practices, risk management, and quality assurance, which are essential for maintaining high standards throughout the manufacturing process. Finally, we have the regulatory expectations set by agencies like the FDA and the European Union, which specify analytical control requirements. These standards help us verify that our water systems meet the necessary safety and quality benchmarks. Overall, these references form a framework that supports the integrity of our water systems, ensuring that everything we produce is safe and effective for patients. They're interconnected, helping us uphold the highest quality standards in pharmaceutical manufacturing. Let's talk about the water quality for HPLC, specifically WFI, or water for injection, which is classified as chemical-grade water, but it's produced through a different process. The water system for HPLC involves several key steps to ensure the water meets the strict purity standards required for accurate analysis. Typically, water starts with pretreatment, where it passes through sand and carbon filters to remove particulates and organic contaminants. Next, we use reverse osmosis, or RO, which is highly effective at removing dissolved salts and impurities. Following that, deionization is performed, either through electrodeionization, known as EDI or mixed bed resins, to further purify the water by removing ions. UV treatment at wavelengths of 185 and 254 nanometers is then applied to break down organic molecules and eliminate microbes. Finally, the water passes through a 0.2 micron filtration to remove any remaining particulates before it reaches the point of use. All these components, filters, membranes, UV units, must be qualified through a series of validation steps. Design qualification, IQ, OQ, and PQ, to ensure they consistently perform as intended. Now, when we talk about the types of water used in HPLC, it's crucial to remember that the purity and quality of water directly impact the accuracy and reliability of our results. So, understanding this entire water generation process and qualification is fundamental to maintaining high-quality HPLC analysis. Let's take a closer look at the different stages of water system qualification. First, we have the design qualification, or DQ. This is where we ensure that the system design meets both the specific needs of HPLC and the regulatory requirements. It's about making sure the plan is solid before moving forward. Next is installation qualification, or IQ. Here, we verify that all equipment has been installed correctly and exactly as per the approved design. It's essential because proper installation sets the foundation for everything that follows. Then we have operational qualification, or OQ. This stage confirms that the system operates within the predefined limits. Basically, it's about testing the system under normal operating conditions to ensure it performs reliably. Finally, there's performance qualification, or PQ. This is where we validate that the system consistently delivers water of the required quality over time. Maintaining this consistency is crucial for the integrity of our processes and final products. These stages together form a comprehensive approach to qualifying our water systems, ensuring they are reliable, compliant, and capable of supporting our quality standards throughout their life cycle. Now, let's take a closer look at the critical quality attributes, or CQAs, that are essential for ensuring product quality. First, we'll start with the chemical attributes. Conductivity or resistivity, for example, gives us an idea of the ionic content in the water, which is crucial for maintaining purity. Total organic carbon, or TOC, measures organic compounds, helping us assess the level of organic contamination. pH, while sometimes considered more informative, still plays a vital role in ensuring the water remains within the acceptable range for our processes. Additionally, nitrates and chlorides, if applicable, are monitored because they can impact product safety or process performance. Moving on to microbiological attributes, these are equally critical. Total viable count, or TVC, tells us about the number of live microorganisms present, so we can evaluate microbial cleanliness. Endotoxins, which are toxic components from bacteria, need to be checked where required, especially in products that are sensitive or injectable. Altogether, these CQAs help us maintain control over the quality of our water systems, ensuring safety and compliance throughout our manufacturing process. They connect directly to our overall goal of delivering safe, high-quality products to the end user. Let's take a moment to review the USB acceptance criteria for HPLC. According to USB 1231 and the PW monograph, it's essential that the HPW used for HPLC must meet or even exceed the PW limits. This means that the HPLC method's performance material should align with or surpass the specifications set forth in these guidelines. The key takeaway here is that ensuring compliance with these criteria is critical for maintaining the reliability and accuracy of our testing processes. By adhering to these standards, we help guarantee the quality and consistency of the pharmaceutical products we work with. So, in summary, always remember that the HPW used in HPLC should meet or go beyond the limits specified in the USB and PW monograph, reinforcing the importance of strict quality control. Let's talk about the sampling procedure to ensure accurate and representative water testing. First, when collecting a sample, it's important to flush the point for about 30 to 60 seconds. This helps to clear out any stagnant water and ensures you're testing the water that's actually moving through the system. Next, always use clean, labeled, and sterile containers. Proper labeling is crucial, so we know exactly where each sample came from, and sterility prevents contamination that could skew our results. Avoid dead legs in the system, because these are sections where water tends to stagnate, which can give misleading samples. Consistency is key, so try to take samples at the same time every day. This helps us track trends over time reliably. Once collected, samples should be transported immediately to the lab. Delays can affect the sample integrity and the accuracy of our analysis. Now, regarding sampling points, we aim for representative locations. These include the RO outlet, after EDI or DI units, storage tanks, the distribution loop, especially the return line, and the HPLC point of use, which is particularly critical. It's vital that our sampling reflects the worst-case conditions. This means we want to capture the scenarios where water quality might be at its lowest or most challenging, ensuring our system is robust enough to handle those conditions. By following these procedures carefully, we can maintain the integrity of our water quality monitoring and ensure our system remains compliant and safe. Let's take a closer look at chemical analysis and frequency. As we explore trending methods, it's important to understand that chemical analysis allows us to identify and quantify the components within a sample, which is crucial for many applications. When we talk about frequency, we're referring to how often certain chemicals or patterns appear in our data. Tracking these trends over time helps us spot shifts and emerging issues more quickly. By combining chemical analysis with frequency data, we gain a powerful tool to monitor changes, ensure quality, and predict future outcomes with greater accuracy. This approach is especially relevant today because trends in chemical composition can signal larger environmental or operational shifts, making real-time analysis more valuable than ever. So, understanding these key points helps us stay ahead, adapt faster, and make more informed decisions based on the most current data trends. Now, let's look at the microbial count calculation. In this case, we've observed 3 colonies, or CFU, which stands for colony-forming units. The sample volume we used was 1 milliliter. To find the concentration, we divide the number of colonies by the sample volume, so 3 CFU divided by 1 milliliter gives us 3 CFU per milliliter. This result is well within the acceptable limit, which is less than or equal to 100 CFU per milliliter, so we can say it complies with the standards. This calculation is a key part of microbiological analysis, as it helps us determine whether the microbial levels in a sample are within safe and acceptable limits. Consistent monitoring and analysis like this are essential to ensure product quality and safety throughout the process. Let's talk about how water quality can really impact HPLC performance. Poor water quality can lead to several issues that affect the reliability of our results. For instance, it can cause increased system pressure, which may stress the equipment and lead to potential failures. It can also result in baseline drift, making it harder to accurately interpret the data. Another common problem is irreproducible retention times, meaning our peaks might shift from run to run, reducing reproducibility. We might also see peak splitting or tailing, which compromises the resolution and quantification accuracy. Lastly, detector contamination is a serious concern because dirty water can introduce impurities that interfere with our detection sensitivity. So this is why water qualification is so crucial. Ensuring we have high quality, pure water directly supports analytical reliability, giving us consistent, accurate, and trustworthy results. Ultimately, maintaining water quality is fundamental to the integrity of our entire analytical process. So, to wrap things up, I want to emphasize that highly purified water really is a critical component in our analytical work. It's not just about having clean water. It's about ensuring the accuracy and reliability of our results. Proper qualification of the water system guarantees data integrity and helps us stay compliant with regulatory standards. Remember, regular sampling, testing, and trending are mandatory practices. These steps allow us to monitor the water quality over time and catch any issues early before they impact our analyses. And of course, compliance with USB, EP, and WHO guidelines is essential, especially for HPLC laboratories. These standards help us maintain consistency and credibility in our testing processes. And I'll leave you with this. Good chromatography really does start with good water. It's the foundation of reliable, high-quality analytical results. For further support, please feel free to contact us. We're here to help with any questions or additional information you might need. And with that, I want to thank you all sincerely for your time and attention today. Your engagement truly makes a difference, and I appreciate your interest in this topic. Remember, we're always available to assist you as you apply what you've learned. So, thank you once again, and I look forward to connecting with you soon.